Okay. Well, good afternoon, everybody. I'd like to tell you about an asset that we have for this condition, NASH. I wanted to go back one if that's possible. This is my second slide. Thank you. So NASH, as many of you will know, actually is the most common liver disease in the US and has no approved therapy, and the market size is actually extremely large currently and projected to be even larger by 2026. And we're developing a novel compound that we've labeled NT7314 as a single first-line treatment for NASH. So just briefly about steroid inflammation, because it's not intuitive, and the general points I'd like to make is that after any form of tissue injury, this is obviously trauma, and this is a myocardial infarct, this is followed by a sterile inflammatory infiltrate. This is a ubiquitous response and is due to cell death and stress. It's mediated by a number of receptors which will be familiar to you, known as TLRs. And importantly, we're dealing in this situation with metabolic syndrome where there's inflammation systemically, but importantly, the amplitude is highest in the liver. So people with metabolic syndrome are not walking in with clinically evident encephalitis or nephritis, but they are walking in with clinically evident hepatitis, which is what we call NASH. And in addition, this inflammatory response is very malleable in the liver. So when patients lose about 10% of their weight, actually it improves very quickly. So we know the regulatory pathways are still in play here. And in the liver, as you know, NAFLD, which is steatosis, is affecting an incredible percentage of the population, and NASH also a very large percentage of the population. And the characteristics are that it's due to metabolic stress, and the inflammation exacerbates the metabolic stress, and various TLR knockout mice are actually protected, and the target for this drug actually is a TLR molecule. So if we look at the development of NASH, obesity, insulin resistance results in steatosis, metabolic stress, and inflammation. And crucially, inflammation is the key to the development of fibrosis and scarring, which actually causes the injury in NASH. In addition, the inflammation actually worsens the stress and the steatosis, and the agent that we have actually blocks inflammation directly. Some of its characteristics are it has a unique mode of action, it blocks inflammation before fibrosis occurs, and importantly, it preferentially accumulates in the liver, it's taken up into endosomes, and unlike a number of other molecules similar to this, does not need to go into the cytosol to be effective. So more about this particular compound. This is a large molecule inhibitor of a toll receptor. The receptor for this target is required for steroid inflammation in NASH, and the therapeutic modality is uniquely suited for NASH because it concentrates inside liver cells. It has excellent in vivo efficacy in models of high-fat diet. We have NOEL in rats and also in primates. And crucially, we actually have safety data in 30 healthy and 52 patients with lupus in phase 1 and phase 2b clinical trials over a period of 8 weeks, and it's entirely safe in these individuals. There's a strong IP position with 7 issued patents extending up to 2030, and we are currently near an inflection point of IND submission. So the hallmarks, as I mentioned, are that it reduces NASH in a high-fat diet model. And I'll just show you one brief data set. So when mice are put on a high-fat diet for 12 weeks, the serum ALTs, which is a marker of liver injury, goes up. There is steatosis and inflammation, and all of these are reliably reduced by the use of this agent. So the application would be in NASH as primary treatment or first-line therapy. However, since it works separately from other drugs that are in development, it's also a candidate for combination therapy. Adults and children above ages of 11. Single primary treatment as first-line therapy and possible combination, as I just said. The dose that I've showed you safety data for, we actually believe it'll be effective for NASH at a lower dose than that, and we want to do animal studies because it concentrates naturally in the liver. So we want to do dosing studies for below the dose that's currently approved in NASH. In addition, it'll be weekly dosing, subcutaneously, and importantly, this pathway really is only switched on during injury, and so the side effect profile is excellent, and we have human data for this molecule. So really, we're actually ready to do a gap analysis of the data set for the clinical trials for this. We have the in vivo studies that need to be done, and then we're basically ready for pre-IND meeting with the FDA by the end of this year. And again, just an overall summary, it's a new disease. This is a promising drug candidate for NASH. We have efficacy data in disease models, safety data in humans, and clear set of steps for IND submission. And in addition to these important developmental steps, of course, this will result in development of this drug to provide therapy to a large number of people who are currently developing liver cirrhosis and liver cancer. Thanks.
Questions? Yes, up here on the aisle. said it's a TLR inhibitor. What specific step in the pathway does it inhibit?
So it basically binds a TLR and blocks binding of ligand, natural ligand.
All TLR?
No, it's a specific TLR antagonist.
Other questions? Amanda?
Yeah. Have you compared it head-to-head against OCA or elafibranor or the GLP-1 agonists?
Not in animal models, but the response is very strong in mice to the point where there's almost normalization. The inflammation essentially goes down to zero, and the steatosis and the other aspects improve dramatically.
Could you just summarize the team?
The team, sure. So, of course, there's my laboratory, which provides some of the basic science. There's the company that originally developed this molecule for an indication in lupus, and they have an active IND in lupus. They pursued, and it actually gave an in vivo signal in lupus from blood cells that were taken out. They didn't further pursue it because it doesn't localize to lymph nodes, which is where they would need it to localize in lupus, but obviously localizes in the liver. So there's the whole team from the company there, and then we've also hired we have internal experts, and we've hired regulatory OCR experts as well.
Fantastic. Well, thank you very much.